Welcome to Chapter 5 of this massively open online course. My name is Stefan Schimura from the Helmholtz Center Dresden-Rossendorf, where I work on the analysis of material surface reactivity using, among others, vertical scanning interferometry. Following up on the previous chapter, I want to illustrate the use of the method and the potential data analysis using the example of the erosion and remineralization of human teeth. Every day, our teeth are subject to chemical processes that can either erode the calcium phosphate-based apatite structures of our teeth or remineralize the eroded structures by providing calcium and phosphate for rebuilding. The erosion is triggered by acids from food and drink or produced by bacteria from sugar. Remineralization is supported by your saliva and calcium and phosphate-containing foods such as dairy products. We have used vertical scanning interferometry to investigate the processes and the enhancement of the remineralization using calcium caseinate protein powder, which is derived as a byproduct from milk production. The calcium phosphate clusters contained in its structure can be used to rebuild eroded dental material. Our samples were longitudinal cuts of human teeth that were demineralized by immersing them in an acidic buffer at pH 4 and remineralized by placing them in a remineralization medium consisting of native saliva and calcium caseinate gel. After each step, the topography of the sample was measured using vertical scanning interferometry. From the measurement, we obtained height data from the same field of view on the sample. This height data was then transformed into roughness data using the so-called SQ parameter, which is the standard deviation of the height data. For every pixel of a given height map, the SQ value was calculated within a 7 by 7 pixel window, giving us a detailed roughness maps, which could be analyzed according to the structure of the roughness distribution on the sample. As can be seen here for the remineralization of dental dentine, the roughness as given by the average SQ parameter of the surface decreases with remineralization time. This is also readily seen in these 3D representation of the roughness maps. Looking at the histograms, we can identify an asymmetric narrowing of the SQ histogram, indicating a heterogeneous surface reactivity as rougher features react stronger to the remineralization process, indicating the successful remission of erosion-induced damages to the tooth. However, we also see that only a small etching steps, as indicated in red, can again reverse the successful treatment. Using this kind of analysis allowed us to track the development of the tooth surface throughout a variety of treatments, from polishing, demineralization, remineralization to fluoridation. From this simple experimental series, using the facile and versatile method of vertical scanning interferometry, we could draw several conclusions concerning dental health. We see no remineralization using only water in the remineralization medium. Saliva proteins play a crucial role in the remineralization process. The demineralization rates are 3 orders of magnitude larger than the remineralization rates, making a longer-term treatment advisable. The remineralized material shows similar demineralization rates as the original tooth material, indicating the growth of biomimetic hydroxoapatite. Fluoridation of the tooth surface successfully inhibits demineralization, but also remineralization due to the formation of a fluoroapatite calcium fluoride passivation layer. The inhibition of the remineralization can be overcome by fluoride addition to the remineralization media, leading to the formation of a fluoroapatite calcium fluoride during the remineralization step. With this, I hope to have illustrated the possibilities of using vertical scanning interferometry for the analysis of the heterogeneous development of a reacting surface with nanometer precision. Similar analysis can be done for almost any surface, from biomaterials, geomaterials, plastics to metals. in order to investigate chemically, biologically, or physically induced degradation processes. With this, I thank you for your interest in this course and hope you stay around for the next chapters introducing even more interesting methodologies. For further information, please check the website and our social media. Thank you.